Hello, my name is Slava Karolis. I'm from King's College London, and I'm also representing the Development Human Connectome Project. And I would like to talk about emerging patterns of functional connectivity in the fetal brain. So thank you for tuning in. And I have no disclosures to reveal. I would like this talk to have a methodological angle, which means I will not give a deserved credit to many pioneering studies, such as mentioned here on the slide, which really provided us with a foundation for understanding the development process in the in-utero functional brain, starting from investigation of emerging interhemispheric connectivity, as well as graph theoretical analysis of the whole brain architecture. What I want to do instead is to focus on the problem of how to build functional brain representation from the in-utero fMRI. In other words, if we have small functional features observed as a voxel time courses of the fMRI sequences, how can we proceed to convert them into more succinct and informative characteristics? We typically call them functional networks. And from there to go and build more general brain-wide characterization of the functional architecture. This problem is obviously not unique for fetuses, but what is unique in this context is the context of the in-utero fMRI, presenting many challenges. First, this is a challenge associated with low signal-to-noise ratio and excessive motion. This topic was previously covered by Athena. There's other hindrances, including conceptual ones, such as rapid and dramatic age-related changes occurring during this early period. And the scale of these changes can be appreciated from the anatomical data, such as this spatial temporal surface atlas of the fetal brain, but there is no doubt that no lesser changes would occur at a functional level. And this presents a problem, because unlike anatomy, functional networks are not an observation. It is a model which needs to be somehow conceptualized. And it turns out the meaning conveyed by mainstream approaches of group-level functional networks modeling is in contradiction to this idea of a rapid change. This is because they implicitly presume an existence of a canonical form of a network. If we take a pictorial example where each dot is a fetus, and a typical approach would be to extract some average representation across the whole sample, effectively disregarding the age-related difference in functional connectivity status among individuals. Obviously, nothing stops us from trying this approach in practice. However, what we observe, if we apply it to fetal data, is a collection of maps which are rather poor in terms of network-like properties, typically featuring a single region in each map. As you can see, this characteristic is replicable across dependent cohorts, suggesting a genuine pattern. There's no doubt that part of the problem in finding the multi-regional distributor configuration is the signal property of the in-utero fMRI. Secondly, maturity of functional connections in fetal brain are likely also play a role. But perhaps we could do better if we shift from the mean model that disregard age-related differences to the modeling that actively exploit them, meaning that it would characterize functional networks not as a static, but as an emerging property of the brain. And the framework which we propose does exactly this. It incorporates age-related changes in connectivity directly into the network estimation. We call this framework maturational networks, or MATNets for shortness. It runs as follows. It starts with computing all pairwise correlations between voxels in the brain, forming an individual dense connectome matrix. This procedure is repeated for each individual in the sample. Then the values for a specific element across individuals are collected into the vector, which then models usual ordinary regression with age as a covariate. In this way, we obtain statistics of the age effect for this element, which is then placed onto a corresponding location of another dense connectome matrix, one for all individuals, which we call maturational dense connectome matrix because it encodes all age-related differences in connectivity in the brain. The remaining steps are to factorize this matrix in order to obtain a set of special maps which are independent. And if we look at these maps, when this approach applied to the fetal data, we can see that they appear to reveal much richer network-like properties, including bilateral sensory motor network number 7, which existence is anticipated but couldn't be demonstrated using group ICA analysis. Furthermore, as shown in this example of the pairs of anatomically related maps obtained using two methods, MatNets demonstrate a superior performance in terms of anatomical specificity. Therefore, the story so far is that MatNets present an alternative representation of functional networks in the fetal brain, and it conceptualizes it as an emergent property and they appear to outperform group ICA in certain aspects. However, can we go further and use this framework to build a brain-wide representation of maturational relationships? In order to do this, we need to make a step back and consider how the MatNets could be interpreted from the computational perspective. It needs to be noted that numerically, MatNets is nothing but just a compression of maturational dense connectome subject to the constraint that they are spatially independent. In the meantime, within maps, the voxels become fused into particular MatNet owing to the fact that they are share emerging connectivity profiles with each other with respect to the rest of the brain. In other words, they develop connection to the same target, and therefore we can ask what these targets are. We can easily contrast them by regressing MatNets against the dense maturational connectome. The result of this regression is a matrix over the beta coefficients, which is a sought-after emerging connectivity profiles, one for each MatNet. And here we can look at this matrix from the two independent perspectives, related perspectives. One is due to the fact that unlike MatNet, which are spatially independent, these profiles are not, which means we can compute a correlation between each pair of rows of this matrix and form another matrix, and we can call it maturational connectome that would summarize this maturational relationship between MatNets. This matrix can be embedded and clustered in order to visualize these relationships. If we do this, several observations can be highlighted. First, the MatNets that are homologous to each other across hemispheres tend to be clustered into the same group. This finding is complementary to the earlier finding by the Marai and Thomason showing increasing interhemispheric connectivity during the fetal period. However, this time we show that the homologous regions not only develop connection with each other, but rather form connection to similar targets throughout the brain. Next, we can observe that maturational connectome contains two central groups of networks, which we can tentatively call perception and action groups, as well as three peripheral groups, which we can call by their anatomical location as ventral occipital temporal, ventral orbitofrontal, and dorsal sensorimotor. The second perspective on the emerging connectivity profile is due to the fact that they are nothing but a collection of special maps complementary to MatNets. Therefore, we not only can look how similar the maturational profiles between mats are, but what is similar between them. In other words, we can utilize the clustering into groups from the analysis presented in the previous slide and extract some averaging measures within each group, for instance, by taking the first component in each group of these complementary maps. This would give us areas to which several MatNets tend to establish connection in age-related manner. We call them maturational hubs. Here is the result of this analysis. Firstly, it shows that the maturational hubs for the two central groups of MatNets, action and perception, contain a significant vascular component as evident by the overlap with the circle of Willis. However, they also clearly contain distinctive cortical areas. For perception groups, the most prominent are the sensorimotor and adjacent parietal regions, as well as bilateral cerebellum. For the action group, these are peri-insular and superior temporal areas. For the peripheral maps, the situation is as follows. For sensorimotor group, the map of maturational hubs is not very distinct, perhaps because these areas as themselves act as a hub for the perceptual group. For ventral orbital group, the main targets are located in the prefrontal cortex as well as a non-negligible parietal component. For the ventral occipital group, the hubs are located within parietal regions, suggested an early integration of the ventral and dorsal visual streams. So, to summarize our findings, this can be as follows. At a methodological level, we present a framework for characterization of functional brain architecture as an emergent property, which apparently outperforms state-of-the-art method in terms of anatomical specificity and richness of connectivity profiles. But also, there are some interesting findings at the system level, revealing how the fetal brain actually emerges, functional connectivity in the fetal brain emerges during this period. Firstly, we find that contralateral matness tend to develop connection to the same targets, but not necessarily to each other, as earlier shown in a work by Thomas and al. Furthermore, the bilateral dorsal sensorimotor and adjacent parietal cortex, as well as bilateral cerebellum, appear to be acting as a maturational hubs for the regions which in adult brain are engaged in perceptual processes. And finally, we observe three types of dorsal ventral integration between functional systems. One is posterior between ventral and dorsal visual stream regions, and two anterior between action and temporal and peri-insular regions, and between ventral, orbitofrontal, and dorsolateral and prefrontal cortices. And I would want to acknowledge the team of the Development Human Connectome Project, without which this work wouldn't be possible. Thank you for your attention.